Great, thanks, Matt. It's my pleasure to introduce Naoki Abe, who's going to be giving the next plenary talk. He's a distinguished research staff member and senior manager in the Artificial Intelligence and Blockchain Department at IBM Research in Yorktown Heights, New York, where he's been since 2001. He received his SB and SM degrees from MIT and his PhD from UPenn. And much of his current research is focused on applying artificial intelligence to agriculture, including the TERRA project, which I think he'll be focusing on in his talk today. All right, Naoki.
Yeah, thank you very much for the organizers for the invitation, and I hope that we can add some little contribution from the industry. I think you will get a lot more tomorrow, but let me see if I can start. So I'm going to be talking about the machine learning approach to both phenotyping and genotype-phenotype association. As we worked on in the TERRA project that was funded by ARPA-E, as some of you here at Illinois know very well, because one of the major teams in the TERRA project was centered at the University of Illinois. We happened to partner with Purdue University to work on this project. And here's a long list of contributors. A block of them from IBM Research. The first three are the main contributors to this work. And there's some excellent interns we got working on these projects, and also a list of people from Purdue University who contributed to aspects of this work. And finally, some block of people from IBM that did less work. Okay, here's the overview of my talk today. I'm going to give just a short overview of the TERRA project itself. I may be preaching to the choir for some of you, but for those of you who may not be familiar, I'm just going to give a very brief overview, especially about the project that Purdue and University of Queensland and IBM Research took part in. And then there are two parts to the technical content. The first part has to do with the machine learning work we did for automated phenotyping, and a particular phenotype that we pay attention to. And the second part has to do with also the machine learning work that we did for genotype-phenotype association, where we developed a novel approach or algorithm to do this simultaneous learning and by clustering, and as it's applied to multi-response GWAS and trait prediction. So let me go on to the overview of the project itself. TERRA stands for Transportation Energy Resources from Renewable Agriculture. It's largely about developing an automated sorghum phenotyping and trait development platform, whose goal is really to develop an automated high-throughput system for determining how variations in the sorghum genome impact field performance, agriculture productivity, and energy potential as biofuel. So the last part, energy potential as biofuel, is what justifies ARPA-E, which is part of Department of Energy, to sponsor this project. There are two main areas of technical challenge. The first really has to do with automated phenotyping from sensor data using ground-based mobile and airborne platforms, as this has been a bottleneck in advancing plant breeding. The second area of challenge is the integrated analytics on high-dimensional genomic data and multi-modal and noisy field and image and sensor data. This is a major technical challenge as well. And the partners and the duration are written. And these two challenges, areas of major challenges, actually correspond to the two topics that I'm going to describe. And they also correspond to what they called Category 3 and Category 4. in the overall TERRA project. So category 1 refers to just the overall, and I believe the University of Illinois and Purdue were two of the few teams that actually got the funding for the whole category 1. But category 1 is then divided into 3 categories. And category 2 has to do with the actual design and building of high-throughput automated sensing technologies. And this was entirely done by Purdue. We took part from IBM Research in category 3 and category 4. And category 3 really is about automated phenotyping from sensor and drone data. Category 4 involves genomic data, and that corresponds to the integrated genotype-phenotype analysis. So 2 of the major technical challenges from our perspective. I mean, of course, category 2 is a major aspect, but we didn't take part in it directly. The 2 major challenges correspond to these 2 categories. So let me then go on to talk about category 3, automated phenotyping, and in particular for counting what's called panicles in sorghum from UAV imagery data. So as many of you may know, sorghum is originally a cereal crop, but it's very well suited for use in biofuel. And the panicle is the head of the plant that holds the grain. And so it is an important phenotype that's related closely to a lot of other phenotypes, including in particular biomass. And we try to estimate this phenotype using aerial downward-looking images taken by UAVs. And we count the number of panicles or estimate the number of panicles in a small row segment of sorghum. Oh, by the way, as you can see there, the sorghums come in various different heights. There are some very tall tall sorghum plants and varieties, and there are some others that are not so tall. And some of the genomic makeups of these phenotypes are understood, but there's a lot that's not understood. So automated high-throughput phenotyping. Phenotyping, as you know, is the process of measuring plant traits, and it's very important for plant breeding. And of course, manual phenotyping can be inaccurate and expensive and labor-intensive, so it's very important to try to automate this. As I mentioned, panicle count itself is a phenotype, but it's also can be used to estimate other phenotypes like the productive tiller numbers and the 50% flowering time. And these are both key phenotypes for sorghum. Okay, the TERRA project I mentioned. So this is sort of a pictorial overview of the phenotyping pipeline of which this work is a component. So in the panicle module, we have raw images like what's being shown there, and individual rows are extracted from the raw images, and then we built some annotation tool that allows human experts to annotate the panicles in a limited number of images, so that a machine learning method can learn from these supervised training data. And once the model is learned, you can receive a new image, and it can predict and estimate the panicle counts. And as I explained, the output panicle count can be used to further estimate other types of phenotypes, and they can go into further processes like crop models or GYs. Here's some examples of image data. They come from part of the fields at Purdue they call the hybrid calibration panel, where they had 18 varieties, which were planted on 4 different plots divided into 12 row segments. And they were imaged at 6 different dates, each about 1 week apart. So the important thing is that there's some time series of image data to use for your estimation. 2 out of 12 row segments in each were annotated, and altogether comprising 864 images that were annotated. And we used part of this for training and testing in a cross-validation in a manner, cross-validation manner. Oh, by the way, so you can see at the beginning of the summer, you don't see any panicles, and then you start to see more and more. There's some more detail about which rows were actually annotated, a little more detail about the dates, and then the count of the panicles that were annotated, and so forth. So there's a notion called superpixels that's kind of important to explain. A superpixel is a group of contiguous pixels with similar characteristics, such as color or intensity. And so there are big groups of pixels that are more perceptual and semantically meaningful than individual pixels. But they're still intermediate units. They're not as big as segmentations, image segmentations, like you segment a face or you segment a particle. They're smaller units. And there are methods available in open source, standard methods available that allow you to come up with these super pixels, and we used one of them. So here's some set of example images with super pixels. We provided different granularities. These are bigger than these. What this is useful for is to allow humans to annotate. They can click on super-pixels, and if they think that these are the particles, and if this super-pixel is a particle, then they can click once and then annotate that one of those is a particle, and that would be much harder to do with pixels. So this is a sort of a useful building block. Let me see if I can. So anyway, there's a bit of an animation. This is the annotation tool that we built. It allows you to, people to easily annotate. It's starting out with this big area picture. It allows you to zoom into a specific area, and you're starting to see particles. And you can start annotating. You can actually run a already learned machine learning model to propose where the particles are. The human can go and fix mistakes, and this allows you to, at first you need to annotate enough so that you can train any model. But once you have a model trained, it makes it easier to annotate further for improving the model. We've also developed a crowdsourcing version of this tool, which is running on IBM Cloud with proper data security and password protection and so forth. So potentially this could be used by many people to annotate so that a single person doesn't have to sit there for a long time. Okay. Let me say a few words about the time series aspect. As I mentioned, there are 6 dates for which we have the image data. And here are some examples of how the particle counts vary over time for some varieties. And you see that sometimes it goes down. That may be because of some destructive phenotyping or some event like storms or hails. But agronomical knowledge will tell you that that doesn't happen without those anomalies. So here's one place where we make use of some domain knowledge or the agronomic knowledge to make corrections in our models. So how do we formulate this problem of counting as a learning problem? Essentially, it's a nonlinear regression. You want to minimize the prediction error where the model makes use of this X, which is all of the features in an image that can have millions of pixels. So if you have just a count of the panicles in a given image as Y, then you would probably need millions of images. So that is not going to work well. So we make use of a notion called image density map, where in every part of the image, there's a continuous value that is kind of a proxy to how much it contributes to the count. If you integrate over the whole image, it should integrate to the number of panicles in the image. But the information is distributed to every little part of the image. So you can train this model with yi varying over the entire image. So you would have actually a lot of training data. So here's an example of an image density map of this image here. So you see that the bright parts have higher densities. So for one image, you get this whole image map as the target Y for regression, and that's how we managed to build a decent model without having too many images. When humans annotate, we provided two ways of annotating. One is called the dot annotation, where someone would just put a dot in the center of a panicle. Then it's automatically distributed as a Gaussian density in the density map. Another way that we developed, so it's explained here. Another method we developed is called area annotation, where using those superpixels, the annotator can actually mark a larger area that corresponds exactly to a panicle, as a panicle. So here they're distributed in a more wide manner, corresponding more exactly to the areas where they're actually panicled. So this is all embedded in that annotation tool. You can see the annotated area corresponding to the distribution in the density map. Okay, here's the architecture of our deep learning-based system for head counting or panicle head counting. The first aspect of it is that it is a rather, it's new, but it's a rather standard CNN, convolutional neural network architecture for counting, and we make use of that. But we make a number of modifications and enhancements that make it work well for this problem. One of them is that we include thermal time as part of the input. That's another place where we make use of agronomy. The image density map is what I just explained. We also rotate the training data and so forth to get rid of orientational biases. We also, because of this observation that the particle count should really be monotonic, we make use of something called isotonic regression, which finds the optimal regressor meeting that constraint, that it's monotonically increasing. So all of these things help make the system more accurate. And here's a series of comparisons that we did with existing and competing methods in the literature. One is comparing with what's known as multi-column counting CNN, where you have many versions of the same architecture for different resolutions. It's called MCNN. We discovered that it doesn't really make a difference in our case. It didn't really help, and I think it has to do with the particulars of this problem. The next thing that we compared with is called CSRNet. This is supposedly cutting edge, state-of-the-art deep learning architecture known in computational vision, where it's very deep, and this front end, what's called the front end, which basically captures the features, they have been trained with literally millions of image data. And then you can leverage that. You can keep that model and then train on top of it to customize to your problem. Now, so the vanilla CCNN method, well, not vanilla, even if you add the rotations and isotonic regression, which improves our numbers, and it does, but it's still not doing as well as CSRNet. Now, but this is not the end of the story. The team didn't quit there. So the thermal time, I think you know all about this probably. Again, this is like preaching to the choir, but it's the summation of the degrees above 10 Celsius degrees. So if it's 18 degrees maximum to date, and it has 8. If it's 22, then it adds 12, and then do that over the season. And supposedly it corresponds to the growing stages, development stages of sorghum. So this is another sort of deep source of agronomy, and we put that as part of the input. So that helped, where it says plus thermal. That really helped the performance of CCNN down from 1.28 in error to 1.18, and it's coming close to CSRNet. Interestingly, adding the thermal degrees doesn't improve CSRNet because it's already been sort of overtrained. Finally, we used another additional trick, which is to use the particle detection. We used a similar model to detect the binary detection of where the particles are. And this allows you to sort of focus on the right part of the image. And this allowed you to finally beat CSRNet in terms of predictive error. So anyway, now the point of this is that CCNN that we developed, plus all these tricks that we developed, is much lighter, lightweighted than CSRNet. It hasn't been trained with millions of generic image data, but with the right kind of combination of domain knowledge and techniques, you can actually achieve equal or better accuracy, and it's much, as I said, lighter and more efficient. So that's the sort of the story of the automated phenotyping section. I think these are some future work and ongoing work that we are thinking of pursuing. One thing that I want to mention is that we have been applying the particle counting to the estimation of flowering time, and we've been obtaining some very encouraging results, like we can estimate the flowering time within about 1 day off. Okay, so I don't know how much more time I have, but then let me spend the rest of the time on the other section, the category 4, or machine learning for genotype-phenotype association. So in the overall pipeline, we have done pre-processing and feature extraction, and then it goes into trait prediction given the features, or all the way to GWAS using the predicted or reference traits together with the genomic data. We are applying, we develop methods that can be applied in both of these two areas. So GWAS, or genome-wide association studies, as you may know, have to do with modeling for various, basically phenotypic traits of various varieties. And for each variety, you have a lot of genomic information features like SNPs. And typically, you do this with regression. You estimate one column of these coefficients, and that get multiplied with those SNPs to predict a single phenotype. So if you have multiple phenotypes, you have multiple columns, but traditionally you would do the regression, each regression separately for each phenotype. So that's what this says. And if there are non-trivial relationships between the traits, that could be inferred post hoc by clustering the columns of these parameters, estimated parameters. If they're similar, then you may realize that those traits are related. Now, here we try to go further. And what we'd like to do is, rather than this post hoc approach, when we do the GWAS analysis, in that process itself, we'd like to discover some hidden structure, like the grouping or clusters within the SNPs or within the phenotypes. So, and this is done as part of that process. Okay, so this would take advantage of similarities that exist among phenotypes, as well as relationships that exist among the SNPs to better estimate this, GWAS relationship. And we want to do this automatically. So the theta coefficient matrix would have this checkerboard-like structure where the clusters are discovered on both rows and columns. So breaking this down in terms of mathematical formulation, the part for estimating coefficients is written here. It's known as lasso, where you have the regression, but also you penalize the number of non-zero, number of large coefficients, because you're trying to minimize the loss and the size of your coefficient. So that forces you to have sparse coefficients, which is a good thing for interpretability. And the bi-clustering part has been formulated in this manner, where we're trying to cluster the coefficient vectors, thetas. We introduce a reference sort of like cluster center coefficients called gammas, and we force the gammas to be similar and thetas to be similar to one of the gammas, and that essentially achieves this clustering, bi-clustering phenomenon. Now, as I mentioned, we want to do this all together. We want to do the estimation and the bi-clustering in a single objective to be minimized. So we just put them together. And by the way, there is a simpler version of this formulation where we don't introduce the cluster centers, the gammas. We directly cluster the thetas. But this is less desirable in the sense, it's simpler, but it's less desirable in the sense that the theta coefficients for one cluster are forced to be the same, not just similar. And this is a little bit of oversimplification, but it's easier to solve. Now, so it's a little confusing because I flipped on you, but formulation 1 is the simpler formulation that I just showed you. And formulation 2 is the full formulation that involves these gammas. And this one, there actually exists an algorithm that can be used to solve. For this one, we needed to develop our own algorithm, which is an iterative algorithm that estimates theta with while you fix the gammas, and then you cluster the gammas, and then you go back and forth for fixed theta. And the theoretical breakthrough is that both of these algorithms have been shown to be shown to converge to the global optimum. So there's a theoretical guarantee that this works as well. So this shows what's known as a solution path of this algorithm, where you see the clustering pattern, biclustering pattern of both rows and columns. As you vary what's called the regularization parameter, where you encourage it to be more accurate but less insistent on clustering, or you become more insistent on clustering, and you get different kinds of solutions. Typically, what you do is you do cross-validation and so forth to pick the best solution. We did some simulation experiments to validate the workings of these algorithms. It's a little bit busy, so I'm not going to go into detail, but it suffices to say that these numbers are better when they're bigger. And formulation 1, the simpler, and formulation 2, the full formulation. In general, you can see that full formulation achieves somewhat better results than the simpler one. Now, the two-step method is to do the clustering first and then do regression. So that's sort of maybe a state of the art that people tend to do. As you can see, it's quite a bit worse. So it does pay off to do it as a uniform, as a sort of a global optimization problem. But this is for synthetic data, just to make sure that the algorithms work well. One of the real data analysis that we tried is on multi-response GWAS, where we made use of close to 1,000 varieties of sorghum with 80,000 SNPs, but we reduced these SNPs down to about 3,000 after removing highly correlated and duplicate SNPs. For the phenotypes, we consider these 6 phenotypes. Remember, we are not just interested in clustering the SNP, but we're also clustering the phenotypes. In this case, the number of phenotypes is relatively small, but we have 2 each of height-related and diameter-related and leaf count-related phenotypes. So we run this algorithm, and this is sort of a pictorial representation of the results we got. On the columns, we have the 6 phenotypes. As you can see, in both the simpler and the general full formulations, you can see that the algorithm clustered each of the 3, the phenotypes into 3 groups. This is kind of obvious, but it's working. And it's come up with a similar but different type of clustering on the SNPs, depending on the formulation. Here's a little sample of what the model is saying for both formulation 1 and 2. What's being plotted here on the y-axis are the coefficients, and on the x-axis are the chromosomes. And so both formulations are pointing to similar areas with some differences. And we asked an expert to investigate this, and she said that for example, there are 3 known height or dwarf genes that play a role in the height-related traits. And one of those genes has been picked by our methods. The other two were not picked, maybe because they're not, so as relevant to this population. We don't know, but it seems to be doing something reasonable. But it also hints at the possibility that we could discover things that are unknown. Okay, so there's also another experiment we did with the same methods. It's a different problem this time. It's a little confusing, but it's for phenotype trait prediction given the remote sensing data. And we applied it in a different manner where we actually cluster the tasks and the features, tasks corresponding here to the varieties. So instead of clustering the features and the traits, we actually, by clustering the features and the varieties. And the reason why this is good is because we had 18 varieties, but we only had 6 data points per each. So if you build a model, separate model for each variety, and there are 200, in the order of 200 features, so you're kind of doomed to not have a decent model. But because we can take advantage of the similarities that exist between the varieties, we could actually build a decent model using this type of approach. And this is an example of clustering that the algorithm came up with for the varieties. And the same expert noted that these varieties that are clustered together seem to share some common features. And by the way, we have compared the accuracy of this method with other obvious approaches, like methods that do not take advantage of this group multitask learning, or just building a single model. We do much better than any of those in terms of accuracy. All right, so here's some concluding remarks about this latter half of the work. Some possible extensions that we could tackle. For example, applications to GWAS with time-varying phenotypes. That's a very interesting direction that we would love to pursue. And if there are any other ideas where this type of method could be used, we would love to discuss further. Okay, so finally the acknowledgement to the grant and Andy Linville for some help. Thank you.
Thank you, Naoki. Is there any questions for Naoki? I actually have one.
Okay.
In your first work, I know you're working on the panicles, right? Mm-hmm. How about disease, and what's the next level down? Are you thinking past that for analysis?
A very, very good question. We haven't worked on disease.
Could you repeat the question?
Yeah.
Oh, the question was, we worked on counting panicles, but do we think about the next steps like detecting disease or other types of applications? And yes, I think it's a good question, and we have thought about those things in the sense that this project was about phenotyping, and so estimating the traits was the most important thing. But when this type of technology is of course more widely applicable, and detecting disease, for example, from aerial images, I think that would be a huge application.
Great.
Yeah, we haven't done that in particular, but we would love to try that. Yeah, thank you.
Any other questions?
Yeah, so
We got a mic, Ray.
yeah, so typically in GWAS, there's controlling for population structure and relatedness among the individuals. I didn't see that in the talk, so my question is that part of the model, and if not, why not?
A very good question. you told me about that as well, and it was educational for me. I think the taking care of compounding information, I think, is very important. We didn't explicitly do that, and I think that may be an area where, if we can collaborate with experts like you, and we talked to Alex about that too, I think we might be able to do an even better job. But the hope there was that this type of method could maybe automatically discover clusters of similarly acting SNPs, like as in epstesis, but it might also pick up confounders due to population biases. I think that's something we may need to pay more attention to. So we would love to do further work.
Okay, sure, thanks.
Any other questions? Yeah, Matt?
Thank you. I just wondered what the computational requirements for this analysis were, because most of this stuff gets done on desktops in R, and I wondered if this was requiring an HPC resource.
Are you referring to a particular portion or all of them, or
Both, actually, yeah. But I'm assuming you need HPC for the machine vision stuff, but for the GWAS also.
The machine vision stuff, because it's based on deep learning, it does make use of GPUs and so forth. The second one, we didn't exactly use HPC. It is somewhat computationally heavy. I mean, it's low polynomial time and it's guaranteed to converge. So it's a significant, step compared to, let's say, heuristic algorithm that you don't know where it's going to go. But it's not like a linear time or something. So it is quite heavy. And we haven't done too much in that direction. I think it's something that we may want to pursue too. Thank you.
Thank you.
Hello. Thank you. Excellent talk. I just have a really quick comprehension question for the GWAS section. So if I remember correctly, there was a row penalty and a column penalty. So within the context of maybe like SNPs and phenotypes, what does row and column phenotype pertain to?
Oh, okay. So the columns, it can be anything actually, but I think it's SNPs versus phenotypes. So because we're also clustering phenotypes.
So Naoki, I think the TERRA project is kind of wrapping up, and I think I was just curious, what are the other big areas that IBM's interested in going forward in digital agriculture, like your group and beyond? I
think that's why I'm here, and we're looking for the next big project and collaborations, and I'm pretty excited. It just ended a week or two ago. So we haven't been looking, very extensively, I think, right now. Oh, but this whole area, I think, is interesting. But as we discussed, use of, blockchain data for food supply chain analysis and optimization type of application, I think that's also additionally interesting.
So if I can follow up on Love's question, can you say a little bit more? I know you do a lot of work in machine learning, but you're also part of a broader group working on AI. Can you say a little bit more about what kinds of AI technologies, what kinds of problems you think in agriculture AI might play a role in? I
see. So I mentioned blockchain. That I think that's very interesting. Mm-hmm. Yeah. And sort of distributed. I'm a machine learning guy, so. Distributed and privacy preserving type learning on top of blockchain enabled data. I think that's one interesting area. Another intersection with HPC, I think that's very important. The other aspect is maybe integrating physical models. We have been working on that topic because agriculture tends to involve a lot of environmental spatial temporal data that have some physics in them. So for hydrological modeling or water logging or other types of disbursement kind of modeling, I think the integration of machine learning and physical modeling, simulation, and maybe HPC also comes in there. I think that's an exciting area.
Any other questions? Well, let's thanks to
Oh.
Naoki.
Thank you.